Bismillahirrahmanirrahim, and welcome back to this next video. And this is the 21st video of the chapter 2 of immunology. From the last video, we started our discussion on the basophils, and in the last video, I've told you that basophils they belong to a specialized class of the white blood cells, which are known as the granulocytes, and they are known as the granulocytes because they contain granules in their cytoplasm. I've told you that the basophils they are very important in the allergic reactions and they are also very important for the parasitic infection. Then I told you about the origin and development of the basophils, and I told you that the bone marrow have got the hematopoietic stem cell. The hematopoietic stem cells, along with other cell, give rise to the myeloid progenitor cell, and the myeloid progenitor cell is actually going to lead to the origin in the development of the basophils along with the other granulocytes like the neutrophils and the eosinophils. Then morphologically, I've told you that the basophils they are a round or irregular shaped. They've got granules in their cytoplasm, and they usually have got a two-lobed nucleus. And I've told you that a particular stain, the right Giemsa stain, that is used to stain the cytoplasm and granules of the basophils, and this particular stain gave purple blue color to the basophil to make it distinguished from the other blood cells, particularly the other granulocytes like the eosinophils and the neutrophils. Then I've told you that the granulocytes, the basophils, they contain a lot of contents in their granules, and the important contents that include the histamine, the heparin, the proteoglycans, the lipid mediators, the cytokines, the chemokines, the enzymes, and the growth factors. Then we started our discussion on the first component of the granule, the first important component of the granule, and that was the histamine. And I've told you that histamine is a biogenic amine. It is very important in the allergic reactions and in the inflammatory responses. And I've told you that the histamine that actually works through four receptors, which are known as the histamine receptor 1, histamine receptor 2, 3, and 4. And each of these subtype of the receptors, they are associated with specific physiological functions and distribution within the body. Immunologically, I've told you that two of them they are very important. The first one was the histamine 1 receptor, which are present on the smooth muscle cells, the endothelial cells, and certain immune cells, and they are very important for the vasodilation and the smooth muscle contraction, as well as for the bronchoconstriction. And all of these they are typically associated with the allergic reactions and in the inflammatory responses. Then I've told you that the histamine receptor 4 that is very important in the chemotaxis, the production of the cytokines, and in the allergic reactions. Now, in this particular video, I want to focus on another important component which is present in the granule of the basophils, and that is known as the heparin. If you talk about the heparin, heparin belongs to a particular class of the chemicals which are known as the glycosaminoglycans, and this is the general structure of the heparin. Now, the heparin that is present in the basophils, that is actually a natural anticoagulant, and what the heparin do is that it is going to enhance the activity of the antithrombin 3, and this antithrombin 3 is an endogenous inhibitor of the coagulation factor that we will see in the body. Now, how this heparin works? If you look at the structure of the heparin have got a particular structure which is known as the pentasaccharide sequence. Now, this pentasaccharide sequence it actually have got a negative charge. If you look at the structure of the antithrombin 3, the antithrombin 3 actually have got the positively charged amino acids in their composition, and the negative charge on the heparin, the positive charge on the amino acids present in the antithrombin, that is actually going to lead to their interaction. And when the heparin that interact with this antithrombin 3, that actually leads to conformational changes, and they are very much visible in this particular image. If you look at this one, before its interaction with the heparin and after its interaction with the heparin, so there are conformational changes taking place in the antithrombin 3 when the heparin that interacts with this. Now, this conformational change is actually responsible for the interaction of two of the important factors. One is known as the factor 10A, and the other one that is known as the thrombin. Now this thrombin is also known as the factor 2A. Now this conformational change, it leads to the interaction of the antithrombin 3 with the factor 10A and with the factor 2A or the thrombin, thereby making these two factors inactive. So after the interaction of the antithrombin with the factor 10A and the factor 2A with this particular antithrombin 3, both of these factors, the factor 10A and the 2A, they are going to move into their inactive state. Now, what would be the end result of this interaction? Now, for that to understand, you should know the normal functioning of these two factors, the factor 10A and the factor 2A. So, let us discuss that. If you talk about this factor 10A, this is responsible for the conversion of the prothrombin into the thrombin. This is your factor 2A. So when the factor 10A it is in the activated form, it is responsible for the activation of the thrombin by converting it from the prothrombin into the thrombin. And this thrombin, it is very important for the conversion of the fibrinogen into the fibrin. And the formation of this fibrin is very important for the formation of the blood clots. Now, how this thrombin works, or how this factor 2A works. If you look at this thrombin, this one is the fibrinogen, the inactive form. So when the thrombin, it gets activated by the factor 10A, it acts on the fibrinogen, it removes some of the component from the fibrinogen, the fibrinopeptides A and B, and the removal of the fibrinopeptides A and B, that actually leads to the formation of the fibrin monomer. What that means is you have converted the inactive form into the active form. Now, when these fibrin monomers they are made, they actually interact with each other to give you this fibrin polymer, and by the help of another factor which is known as the factor 13A, you are actually going to make this blood clot. Now, if the antithrombin 3 that is inactivating the factor 10A, that would mean that you will not be able to make the active thrombin. And if you are not able to make the active thrombin, that means the fibrinogen will not be converted into the fibrin. You will not be able to get the fibrin polymer and hence a blood clot. Now, why this is important? Because it prevents the thromboembolic events and it maintains the blood fluidity. For example, if there is infection, there is inflammation in the body, so you want the movement of the immune cell very quickly towards the site of the infection, and by stopping the blood clotting, the heparin is making it sure that blood is moving very smoothly. It is preventing the thromboembolic events, thereby helping in the elimination of the infection at the site of the infection. Another important component that is present in the granules of the basophils, they are known as the proteoglycans, and that will be the topic of discussion of our next video. So if you like the video, please subscribe to my channel, hit the notification bell. If you have any question, you can ask me in the comment section, and do share it with your friends.